Hi and welcome back. In this video, I will discuss the experiments that attempted to crack the genetic code. In order to fully understand where these experiments came from, we must start at the basics. Translation is the main focus of these experiments. It forms a part of the central dogma along with replication and transcription. This is defined as the process in which information is transferred from RNA to a protein through a specific code that determines amino acid sequence. Therefore, it is crucial to understand where they come from. Amino acids are held together by peptide bonds, thus creating 4 different structures. However, in this video, we will focus more on the primary structure since it comes from this amino acid sequence. In 1961, an experiment conducted by Brenner and Crick established that a codon consists of 3 non-overlapping nucleotides. This led to the creation of the triplet code, which determined 3 reading frames per strand along with the start and stop codon at the beginning and end respectively. This became the basis for all following experiments. A notable figure is Marshall Nirenberg, a biochemist who was set on determining the mechanism for protein synthesis as it relates to RNA and DNA. He knew that ribosomes could be combined with amino acids to create proteins, but he wanted to know how and which nucleotides would do the trick. In 1961, he joined forces with one of his fellows, Heinrich Matthey, to develop a new experiment. They were the first to determine that specific RNA is coded for a specific amino acid, all in an attempt to crack the genetic code. In this figure, you can observe the different steps they took. They began by extracting the contents from E. coli cells, except for mRNA. They used homopolymers to determine the specific amino acids. For example, they found that U encoded the amino acid phenylalanine. This process was repeated until they were able to isolate each amino acid. At the end, they discovered newly synthesized proteins. From this experiment, they were able to conclude that basically any RNA can produce a protein. However, soon after, Nirenberg realized that not all amino acids were found this way, which is where Lieder came in. They developed a new experiment where they used short artificial RNA sequences and multiplanar filtration to identify the remaining 61 codons. They were finally able to crack the genetic code. The results of these experiments gave us what is known as a coding dictionary. It is a table that contains RNA codons with all 64 triplets, serving as the basis for experiments to come. This table is determined to be almost universal since it contains codon sequences and amino acid pairings. It is also described as unambiguous, degenerate, contains stop and start punctuation, and has no internal punctuation. Thank you so much for watching and stay tuned for more.